Hi everyone, Leo here, and I'm just going to talk to you guys quickly about and go over with you guys the the methods behind TLC for this lab and for this week, as well as then the theories behind those methods, and then also talk a little bit more about the why. Why are we doing this, especially in relationship to the experiment from the week prior to this one, which was CBD solubility. So first, what is TLC or thin layer chromatography? TLC is a method for separation, identification, and purity determination of non-volatile mixtures. It's important here to note that these are non-volatile mixtures, because if they were volatile mixtures, then they would vaporize readily in room temperature, which would mean that we wouldn't be able to do any assays with any liquid eluents as we do here in this lab. So the very first step for TLC is the preparation of the actual plates themselves. And the TLC plates here are made out of silica, but they have a silica side and a plastic side, which is important to note when you're laying these down and cutting them and whatnot, because you want to make sure that you're laying them on the plastic side and that when you're actually going ahead and spotting the plates, that you're spotting the silica side. And a good rule of thumb in terms of cutting is to cut them at about 4 by 5 centimeters, in order to ensure that both A, that the plate fits in the chamber, and that B, that you have enough space on the actual plate itself in order to do the spots that you need to do. So after you've prepared the plates, you're ready to go ahead and actually spot the plate. Before you do that though, you have to clean the TLC capillary before and in between uses. And you can do this with either acetone or water. And all you do is you just dip the capillary into the water or acetone and then do fake spots, for lack of a better term, on the paper towel itself about 2 to 3 times to ensure that whatever else was or could have been inside of the capillary is no longer in there and won't affect your experiments. And then I wanted to point out also that in this particular week's experiment, we performed the spotting method, but there's also another method called the streaking method. And up here you can tell and see a little bit quite obviously that the spotting method is when you spot on the line, when you actually spot your analyte, whereas the streaking method is when you use streaks instead of your analyte. And the difference here is that you do a one long streak with, let's say, this red substance, allow that to evaporate, and then do another streak with this, let's say, purple substance, which then the eluent would separate as it moves upwards, making these separations here. And we're going to talk about the theory behind this over here in a second. But first, I want to talk about the stationary phase, mobile phase, and the analyte over here. So after spotting the plates, you're ready to go ahead and get your eluents into your TLC chamber, and then put your TLC plate into the chamber with the eluent ready to go. And so with this combination, we get the stationary and mobile phase as well as the analyte. And the stationary phase is the silica on the plate itself. This is the phase that doesn't move at all in this experiment. The mobile phase, the phase that actually moves, is the eluent. And in this experiment, our eluent was 80% hexanes and 20% ethyl ether. And then the analyte for any TLC experiment will be what substance you're actually spotting. So it would be the water, methanol, ethanol, CBD, and so on. The different analytes are what you're spotting in the different spots that you're making. So after you've spotted, you've put everything in the TLC chamber, you're left to wait until the solvent front moves up towards the end of the TLC plate. And what was pretty cool about the videos on Blackboard is that you could actually see the solvent line move along the TLC plate. Here I drew it out and I just kind of drew the boundary here as it moved from this position to the end position over here. And this happens through the same means of at which the actual solvent moves up the capillary, through a phenomenon called the capillary action in physics. You won't need to know exactly what that is for this class, but it's just good to know that capillary reaction, when fluids move up condensed spaces, this is what allows the solvent front to move up the TLC chamber and allows the samples to move up the TLC, or I said chambers, the TLC plates, and move up the TLC capillaries. And so after you've spotted, put everything in the chamber, after the solvent has moved up, um, or eluent, sorry, not solvent, has moved up the TLC plate, you can take the TLC plates out, allow them to evaporate, and then go ahead with the visualization studies. So for our visualization for this week, we did the UV lamp, the I2 chamber, and the fast blue dye. The UV lamp is composed or allows visualization of multiple bonds within the structures that you're looking at, whereas the I2 chamber allows for visualization of hydroxyl groups within the structure that you're looking at. And then the fast blue stain allows for visualization of cannabinoids. So you can start imagining the types of structures you could see um, under a UV lamp versus the I2 chamber, and then versus obviously the fast blue stain, which is cannabinoids, or in combination, right? And start to kind of postulate what structures would you see under which visualization methods. So now on to the fun part. So what is actually going on in this experiment as we are conducting it? What is actually causing these different spots to form? So when we look at the chamber and the plate, this is my poor drawing of a plate at side view. We can see that the stationary phase, or the silica, is polar, versus if we compare that to the eluent, which is ethyl, forgot an L there, ethyl ether plus hexanes, is nonpolar. So we have a mobile phase, which is nonpolar, and a stationary phase, which is polar. And this sets us up perfectly to be able to create spots of different separations or different lengths from each other based on this concept of like dissolves like, right? Because the stationary phase doesn't want to hang out or dissolve with the mobile phase because they're not alike. One is polar and one is nonpolar. And so what happens here is, if we look at the experiment we conducted, we can see that. Now remember, here when it says water, methanol, and ethanol, there's presumably also CBD with that combination. So we can see here that, first of all, CBD is nonpolar. We just said that our mobile phase is also nonpolar, whereas our stationary phase is polar. Like dissolves like, so the CBD standard would be expected to be towards the top end of the TLC plate as it travels with the also nonpolar mobile phase, the eluent. Now, when we look at methanol plus CBD, for example, or even ethanol plus CBD, for example, methanol and ethanol are relatively polar compared to CBD, and also relatively polar compared to our nonpolar eluent here. So what happens here is that this methanol and ethanol, since they're so polar compared to everything else, they're going to want to stay with the polar, stationary phase. And so because of that, they're not going to travel that far in comparison up the actual plate. Whereas the CBD, being nonpolar, being which is in similarity to the eluent, will actually move up with the eluent with the mobile phase and move farther up. And so this is important to know moving forward in terms of your SIs and your IMFs and how it affects the experiments you're running, and in particular your TLC experiments, and how then using this information you can start to kind of postulate and kind of predict uh, where spots will be based on their polarities in comparison to the eluent and the the stationary phase versus the mobile phase. And why are we doing this? Just to wrap it up, Why are we doing this in comparison or in contrast to last year or after last week's lab? And the reason that for that is to actually confirm and see whether or not these uh, solvents were soluble for CBD, right? Because if they were soluble for CBD, we'd expect that liquid portion, right, of these vials right here, we'd expect that liquid portion to have CBD molecules, and thus we'd expect to see, remember at the very beginning, separation of CBD plus that solvent, and then thus identification of CBD. So TLC in this week is allowing us to then test our solubility experiments from last week and see whether or not we were able to dissolve CBD in methanol or ethanol water or any other of these solvents based on the fact of whether or not there was or wasn't a spot along with that solvent that is at the same length or distance in comparison with the CBD standard.